obvious out of this is to develop biomarkers early and discovery and target validation with the idea of getting hits out that are ready for the optimization stage to be further developed. In the optimization stage, we're going to talk about today, I want to focus in on what's going to be required under this program. And this program is really designed to do optimization of small molecules and biologics. And also, we want to see a component of biomarker, target engagement biomarker work so that we can see an early read. And we'll talk more about that. The goal here is to get to the IND and in first in human studies so that we can hand this off to phase 2 an EPICNet or some other program in order to continue the clinical studies, because we definitely want to keep moving the project forward and in eliciting investment from outside interests. So the new title of the FOA is going to be Non-addictive analgesic therapies, therapeutics development for small molecules and biologics to treat pain. It will be clinical trial optional, unlike the previous FOA. The goal is to accelerate and develop novel, non-opioid, non-addictive analgesics. Our 5-year benchmarks on this is going to be hopefully having about 5 projects that with appropriate assays, models, and tools ready for preclinical optimization from the early stage, as well as at least 3 projects in the IND and human safety stage from the later stage that we're talking about today. So the optimization program is going to be under an RFA. The number has not been released yet, so I can't give that to you, but hopefully it will be very shortly. I expect it anytime in the next week or 2. It'll be a milestone-driven program, and it'll be cooperative agreement, meaning substantial NIH scientific involvement. The idea is to identify and conduct the studies necessary to meet the requirements for initiating optimization and/or IND enabling studies. And the optimization, identify a development candidate, move that development candidate through IND enabling studies to phase 1. We're going to hopefully provide resources, and I'll talk a bit more about the contract resources available to you. The end goal in all cases is, as we discussed, move the project through the pipeline, seek partnerships, and get it ready for phase 2. So those of you who may be familiar with me from the Blueprint Narrow Therapeutics Network will recognize this slide. We plan on setting up similar set of resources available for both small molecules and hopefully for biologics as well, as we're expanding that realm. So we have in place from the Blueprint Narrow Therapeutics Network contract suite that includes things like medicinal chemistry contract, a relational database system to if you're doing optimization in small molecules and potentially in peptides and other things that may be of utility to you. We have contracts in place for PK and toxicology, which can be utilized, and it typically is definitely utilized for large animal PK and the GLP toxicology studies. We do have drug manufacturing formulation for small molecules. We do not have any contracts to date that can handle biologics, but again, those are things that we envision that we will have down the road for this and other programs. Our PK tox can handle biologics, as can our clinical trials and our phase 1 clinical trial units here, focused mainly on healthy volunteer studies. And this is really first in human, SAD, potentially MAD studies. The idea is that it'll be a 2-phase mechanism. It'll be a UG3, UH3 mechanism. And so therefore, we expect to launch up to 17 projects in the first 3 years, probably around 5 in the first year of this program, to move this project forward and get a pool of projects. We do expect significant attrition at the stage of moving into the UH3 phase, and we expect maybe 5 projects will advance to the UH3 phase and have access to the larger toxicology contracts and things like that. And this will be based on a number of criteria, which I'll discuss later, but it includes funding availability, but also includes how much progress we're making on the optimization front, as well as the biomarker development front on things. We do anticipate the first receipt date to be November 18th, with dates continuing through June of 2022. As I mentioned on the biomarker side, we do want to see that you put together a team, and that team includes people on disease biology, and includes people with the ability to hopefully develop these PD and safety biomarker things, to hopefully be able to interpret IND toxicology and predict our human dosing paradigm. And I think that'll be quite useful and make the project much more attractive to investors. The overall goal, as I've said, is to optimize and accelerate the whole process. You need to come in with a rigorous biologic rationale for the intended approach, so that early stage work that Michael's still setting up that program on will be a lead in for future projects, and maybe in year 2 and 3 they may be hitting us from some of those. But you all that may be just ready right now, you need to have that biological rationale in place for your intended approach. You need to have a promising small molecule or biological starting point for the optimization program, and you need to have scientifically sound assays to optimize and test the reagent. And that's the one thing that the NIH contracts can never do for you, is provide you the disease biology assays. And so when you develop your team, make sure that's part of your team. You need to be able to do the assays to optimize your primary, secondary assays, your in vivo assays. We have presently 9 ICs plus the Office of Research on Women's Health signed on to this FOA. All the grants will be held within NINDS, and because they're a cooperative agreement, we hope to pull science officers from these institutes for programs within their missions. The key resources that we have available, I talked about in that previous slide, include the contracts, but they also include subject matter experts. And we put together a lead discovery team around each program. That lead discovery team is tailor-made to that program and includes subject matter experts that are appropriate for this phase of the program you're in. So if you're in an early stage program, you might have simply a med chem consultant and a DMPK consultant, but as the program would progress, you would have a toxicology consultant, a regulatory consultant, a CMC consultant, a chemical process consultant. Likewise, putting that same kind of sets of consultants in place on the biologics front, and Anne Marie is working on that as part of her mandate on the group, as not only for this program, but for BPN Biologics as well. So in addition to that, because this is, as I said, a collaborative program across NIH, you have the opportunity, if you choose, to work with NCAT's intramural scientists and programs. But the caveat there is you need to reach out to establish a CRADA with NCATS prior to submission, and that takes a while. So if you were thinking that and you want to come to November, you need to sort of reach out right away to NCATS and have a conversation with them and start that process. You can additionally create your team any way you see fit in terms of bringing your own expertise or working with us. You know, additional IC resources may be available. If you're aware of them and you can tap into them, then by all means, you can propose to do that. That leads me to the sort of the overall entry phase. Everyone comes in at the UG3 phase, and we anticipate most projects will probably enter at the discovery phase, mainly due to the fact that having the package of data for development compound requires that you have a complete package. Everything, you have dose range finding tox, you have it all done. So the reality is most people are coming in at the end of the optimization phase, and they need to do a number of things before they can really declare their candidate. So most people will come in at the discovery phase. Now, along the discovery paradigm, you could potentially say, I just have a hit, or that I have more advanced hits and I'm ready for lead optimization, and we can discuss that. And I think the most important thing to take away from this is discuss your budget with the NIH staff. The UG3 is expected to be 2 years and the UH3 3 years, with a maximum duration of 5 years. We will take academic institutions and small businesses, as well as foreign institutions and components, are all eligible to apply under this single FOA. There will not be a U44 version of this FOA, unlike the previous time. So this will just be the UG3 UH3. Activities that are not responsive, that you should know are not responsive, are things like screening to identify hit compounds. That's going to be in the earlier stage. Basic research and studies of disease mechanisms, animal model development of diagnostic and diagnostic devices, studies directed beyond phase 1 clinical testing. So if you're coming in and say, I just want to do phase 2, I'm ready for phase 2, that's going to also not be in our mandate. And the last one on here is really important, and I know it's in the questions already. Opioid sparing projects and projects targeting mu opioid are also not responsive to this FOA. As I mentioned, it's a two-stage mechanism. The thing I want you to take away on this slide is that in order to go from UG3 to UH3, there is an administrative review and we will look at each project based on its merits and determine overall from a portfolio which the best projects we think have the best chance to succeed within the budget we have in order to move things forward. You'll need to have entered all the criteria to come in, execute on your plans, met your milestones on a regular basis. Even to go from year 1 to year 2, we're going to have milestones that are reviewed at least annually and determine whether we will release the additional money for year 2 based on milestone progression. That's going to be true for every year. But as I said, we do expect significant attrition at this early stage based on history of going from UG3 to UH3. I would love to have a problem of having too many great projects and to go argue for more money, but that I will hope is my problem down the road. As I said, the UG3 phase is expected to be 1 to 2 years. Typically, most people budget for a 5-year budget, so it'll end up being 5 years. So end up being 2 years, excuse me. We've already talked about the entry criteria. We encourage projects proposing the optimization activity that they have the preliminary efficacy testing and pain models, and they characterize and have tested their compounds for ADMET. So if you're coming in saying, I'm at this later stage, you need to have some data to back that up. And the overall goal is to move things through. So if you come in at an entry point, I just have a starting point and the biological rationale, we'll move towards developing that as part of the program. If you're coming in and say, I'm already there, you need to present that data. The overall goal is to characterize a lead candidate and ready for in vivo testing and additional optimization that may be necessary. So it's theoretically possible to come in the beginning and have 3 years of optimization going in, followed by 2 years of development that crosses the UG3 UH3. It all depends on when you're entering in the program. The UH3, which will move forward to based on administrative review, any further optimization activities can be completed. We'll obviously go into the GLP and non-GLP toxicology, PK, formulation studies, things like cell bank development and testing, gene expression levels, biodistribution, tumorigenicity and immunogenicity, process development, manufacturing, and eventually the GLP tox, as I mentioned, will all be things that we'll be doing in that later stage. In addition to that, we could, if you propose to do a clinical trial in the application, then you need to fill out all the paperwork as if it's a clinical trial application, and it'll be judged based on that. If you're using BPN contracts for the clinical trial, meaning we're doing the clinical trial through our contract and not in your budget, then it's not a clinical trial application. That same paperwork that you would normally fill out, we would fill out when the time comes to run the first in human studies. I've talked about administrative review. It's going to be successful achievement of milestones, feasibility, competitive landscape, program priorities, and availability of funds. So lastly, I just want to leave you with some things to do and some things not to do. So the most important thing is read this funding opportunity carefully. If you have questions, contact us. It does require certain things like a table to show me what you're going to do yourself and what you're going to do via our contracts. It does require you have a 2-page maximum patent strategy that's in the other attachments, amongst other things. Do stick to your page limits. Don't try to circumvent them. That's a way to get your application withdrawn. Put forth a solid scientific premise with address the rigor. Address the obvious criticisms of your starting points. Because when you don't, reviewers assume that you didn't recognize them. So it's better to say, yes, I recognize this has got a structural alert, or I've got this key issue that is going to be the focus of what we work on. And if we don't achieve it, we won't go forward, but we believe we can because of X, Y, Z. As I mentioned, you clearly have to indicate what's going to be done by the grant budget, what's going to be done by NIH contractors. It's required in the application to create a table. Do include budget sheets for both the UG3 and the UH3. Don't just budget just the UG3 portion of it. Give me the entire budget. Address rigor. Address rigor in the research strategy section. Don't bury it in the invertebrate animal section. It needs to be addressed explicitly, both for your work as well as for the prior literature that you're citing as key to determining whether your project moves forward. Some additional things to do: if you're not registered with all the registrations, start now. It can take 6 to 8 weeks to get done. You're going to be running into the end of FY20, FY21 conversion. Some things will be down during that period, so just make sure we get started early. Consider submitting your application early. The deadline may be, if you give it to me a week before and I see an error, I'm looking for them, I can potentially get you to correct the error. But if it's on the last day of the grant, and then I can't get you to do that. So consider putting it in early if you can. Again, discuss your budget with the NIH staff. If you have questions, reach out to us. We're happy to talk to you about it and get it squared away so you do it correctly. Talk with your tech transfer and your business development folks in advance, because you need to put together that patent strategy. You need to have buy-in for who's going to, or how you're going to pay for the national filings and things like that down the road. Some things not to do: don't plan this to be your sole source of funding for your lab. Because it's a milestone-driven program, because we're expecting significant attrition, projects can end abruptly. And if you can use our contracts and not hire someone, sometimes it's better to do that if you're not in a position to keep them if the funding were to stop. Do not plan the NIH contracts to do disease biology. Do not include the NIH contracts in your budget. Do not reach out to planned NIH contractors for letters of support, because some of these, as you saw on the slide, sometimes we have multiple vendors. They're bid out at the time. I can't tell you which vendor is going to be your vendor. And it's not necessary. So with that, I'm at the questions and discussions. Hopefully I left plenty of time. Perfect. And we can talk about the application process, the review process, proposals and budgets, contracts, intellectual property, all of it. So with that, I will go ahead and pause my screen so that when I pull something up to answer your question, I can go on.
This is Michael. Could I add a few?
Absolutely, Michael.
Thank you. Hi, this is Michael Oshinsky. I'm the Program Director for Pain and Migraine Research at NINDS, and I'm also the co-chair for the preclinical and translational pain research in the NIH Helping to End Addiction Long-Term, the HEAL initiative. For these type of projects, there's really a expectation on the part of the review committee that there's pain expertise on your team. So if you can include that expertise, either in the form of people who have worked in the pain field and developing therapeutics in their area, or including a clinician to guide you in helping you select which therapeutic condition in pain or which type of pain is appropriate to match the biology of your therapeutic, that typically helps out in review. In addition, as Dr. Cywin had mentioned, is that reaching out to NIH staff, either at NINDS or for other pain conditions which may be more appropriate for other institutes, such as musculoskeletal pain, which would be NIAMS, or pain associated with the visceral organs, or maybe a diabetic neuropathy, which would be NIDDK, to reach out to those individuals and make sure that there's sound biology in your application and the way you're framing your argument, and we can help you make sure that that part of the application is focused.
Thank you, Michael. That's excellent points all around, and I should definitely mention, I agree with Michael on, having pain expertise is critical. Having the clinical insight, I think, will really be an essential part of this, and it will help you with that biomarker planning for what's going to help you predict the human dose correctly, what's going to show you target engagement in the preclinical space, and may also help you down the road in what you're thinking about for biomarker development.
So, Chuck, how do you want to do the questions here?
So Tim has questions up on his screen, and Tim potentially could add you to the question. I'll read the questions out, and we can all answer them if there's a thing. So the first question up is the total amount of funding for FY21 for this announcement. And Michael, can I actually say a number? I'll ask you that question. Probably not.
Yeah, at this point, the exact allocation is not fixed, but you should feel comfortable that there's a significant allocation and expectation that there's going to be a number of projects funded in FY 20 and 21.
Well, 21.
Sorry, 21. Yeah, the first set are going to be 21. Is that correct?
Yeah. So okay. We have a receipt date in November. They'll be awarded at May Council, so that'll be FY21. Which starts in
21.
Weeks.
That's right. Yep. That's right.
And as I said, we anticipate launching 5 projects the first year, which means we have to launch more projects the next year in order to get up to that anticipated 17 number by the end of 3 years. So we do have a significant number of projects we expect to launch, and this is a big undertaking to do all this. So keep that in mind. Is it okay if the pain expert for the team is a professor based in Canada?
I can address the question.
Sure.
So when it comes to foreign components in your grant or in your project, so there's work that's allowed to happen outside the United States, and you can have collaborations and expertise outside the United States. At NIH, we understand that team-based science and science that takes the best strengths from wherever the person is geographically is the best science. Okay, but when it comes to the primary institution, although it's absolutely not required, it's easier administratively if the primary institution is in the United States. So to answer your question directly, it's 100% fine and great if the proper person for you to collaborate for the pain expertise is in Canada or in another country.
Okay. So the next question up is well, basically, what about multi-receptors, including mu instead of only mu receptor? And I think if they're hitting mu, and Michael, correct me if I'm wrong, then that is not going to be responsive to.
So not only is it not responsive, but you're going to have to demonstrate that there's little or no abuse liability. And I understand in the case of maybe peripherally restricted mu agonists, or in the case of the mixed agonists, they may have less of abuse liability, but that abuse liability is a very important part of us on the program side deciding whether or not your application is going to be funded. And we're giving you advice on the front end. We're not saying that there's 100% no to your mixed agonist, but it's going to get a lot of scrutiny, that I can tell you.
The next question.
If the data's not there on the front end, that's going to make it harder to make that decision to fund it.
So the next question is pretty straightforward. Will non-mu opioid targets like kappa and delta opioid receptors be considered responsive?
Yeah, that's just fine. Yep.
That's yes. How about a non-specific pan-opioid receptor agonist? I think, Michael, you just answered that question.
Yeah, if it has abuse liability, it's going to be a no.
Does the program look at mixtures of compounds or partially purified extracts from natural product sources, or does it only consider purified single chemical entities? So the answer to that is you should talk to us. It comes down to, are we going to go down the IND pathway to move this mixture to a drug candidate that we're going to test in humans? If the answer to that is yes, then I think we can fit it within this paradigm. And that's really where we have to discuss. Can we propose a small molecule which is already FDA approved for other conditions to explore its application in analgesics for a specific disease? That comes down to the same sort of question I just got. If it's going down the IND pathway, then we potentially can, in fact, do that. If, in fact, it is going down the ANDA pathway, the 503B2 pathway, then it's less likely we can accommodate it within this mechanism. But in any case, in all these cases, we would have to be having a biomarker portion of this program as well coupled in here, and I don't want to lose sight of that. So you would need to be talking about how that your existing drug is, you were able to get to where you thought it was going to be useful in this indication area, and what biomarkers were being developed and along those lines.
Chuck, there's a question about the company being registered in the United States, but having a majority owner outside the United States. And that's related to the previous question, but it has, I think it's hinting at the SBIR or the non-SBIR version here. So I just want to bring that out into the open. So there is not an SBIR version of this announcement, but companies are permitted to apply for this. And they will be evaluated on the same footing as any academic institution or any other institution that are applicants. So if your institution is a registered entity for receiving, grants from the U.S. government, and that information you can get for how to register for that on the SBIR website, then it doesn't matter that your majority owner is outside the United States. That's just fine.
Correct. Now, you do not have to meet SBIR rules under this funding.
Exactly. Yeah, that was the next thing I was going to say. You don't have to meet the SBIR rules, and just for your transparency, the reason why we chose to do the to not put the SBIR component here is because we usually do that to take advantage of that additional set aside. But HEAL Funds has a set aside itself already. So there wasn't necessarily an advantage to have an additional SBIR version of this. So that's why there is an SBIR version, and all the institutions will be on equal footing for getting these awards.
So the next question is, how broad does the analgesia have to be? What if the indications are fairly limited? Well, as you saw from the screen, we have 9 ICs signed on. It could be in any of their missions, and it could be fairly limited, Michael, to my understanding here, as long as we fall within in the mission.
It could be for specific conditions such as sickle cell disease, and it could, the mechanism of action of that entity that you're optimizing can have a specific effect only in sickle cell patients, or diabetic neuropathy, or cancer, chemotherapy-induced neuropathies also. So, the emphasis here is that it has to benefit the pain outcome of that condition. All right. So if it's a disease modifying itself, could be okay, but you have to demonstrate a benefit for pain in the patients. Okay, that's the goal of the project, is enhancing pain management. So yeah, that's going to be an important part of the consideration.
Okay, Tim, the next question, the one that's on the top of the screen. Okay, is a local anesthetic responsive, assuming drug offers real clinical advantages for analgesia where current drugs are limited?
Yeah, that's responsive. Mm-hmm.
Yep.
That would be for acute pain.
Mm-hmm. Yep. I'm an academic researcher and co-founded a small business. If I apply from the university, can the business serve as a subcontract? Yes. It's like any other co-investigator. How different is the FOA from Blueprint Neurotherapeutics Network program? Can we apply the two in the next few months?
Yeah, I think that they could apply to both BPN and this program.
You can't apply to both programs. You can't apply anywhere NIH the same specific aims at the same time under review. So whichever one came in first would be okay.
This is an important no-no that Chuck is making over here. So you can't apply the same project to two different granting mechanisms. Now, after you've received the score and summary statement, then you're permitted to apply. You should reach out to the program official and find out if it's going to be funded, of course. But and if it's considered for funding, you shouldn't. But if it's not considered for funding, once you have that summary statement in hand, you can apply.
But the big difference, I should say, between the two programs from a small molecule, first off, this takes small molecules of biologics. BPN is expanding to biologics, but not yet. The other big difference is Blueprint Neurotherapeutics Network is neurotherapeutics and has a different, although somewhat overlapping in places, set of institutes and centers signed on within their missions. So for instance, for the HEAL, things within the mission of NCI, NIDDK, NIAMS are not part of BPN. So you wouldn't be able to apply, even if you had a pain condition, to BPN under one of those missions, like chemotherapy-induced pain wouldn't be responsive to BPN, but it would be responsive here. Diabetic neuropathy, something addressed at diabetic neuropathy might not be responsive to BPN, but it would be responsive here. So that's where some of the advantages of the HEAL. Plus, it's unlike the BPN, this is an actual set aside. And so there's, a set amount of money going to be funded to this.
If you have a pain project or developing an analgesic, you're better off applying to this program than BPN.
I would think so. Is it okay to propose a development of a compound that a small business is already developing via an SPR, but for a different indication?
So as long as there's no overlap.
Yeah, it would have
to have the same activities on both projects.
Right. As long as you can parse out the two projects, so I'm not paying for things twice, and you can clearly show me that, it's okay. In addressing biological rationale, how important is it to emphasize why your target would be expected to have low abuse potential? Or is it sufficient for the target not to be an opioid? I think at the end of the day, every pain project, you're going to have to demonstrate it doesn't have abuse potential. But would you know that up front? I think in some cases you may not know that, and that's going to be have to be part of your drug discovery paradigm, is evaluating abuse potential early in the program through things like condition-place preference or other models, and eventually
self-administration.
Eventually, you have to get to the gold standard of self-administration models.
Yeah.
What about chemo and radiation therapy pain side effects for intraoral pain, wounds, and inflammation? Pain side effects.
So we're saying to treating the pain associated with radiation therapy, or
that would be fine. That's just I'm not quite sure what it says. Pain side effects. If you're treating pain, it's within the mission.
Exactly. Even if it's a side effect of a therapeutic treatment.
Yep.
That's fine.
Next.
When will the RFA be available? The timeline is hopefully within the next few weeks.
Yes.
The sooner the better from our perspective.
Yes. I expect it any day now, which is why you see the notice saying we're anticipate funding it. Do you consider a novel drug delivery system to enhance pain relief of existing? Do you consider novel drug delivery systems to enhance pain relief of existing drugs? I think that would not be responsive. That would go to the device.
Devices, yeah. I think it's devices, yeah. Unless it's like doing cage delivery or something.
Yeah, talk to us because we may have a home for you.
Yeah, that's right. Just contact us.
Does NA support phase 2 human clinical studies? If so, when is the appropriate time to apply? After IND declaration?
We have an answer for that. Yes, we do.
During phase 1. The appropriate time, well, part A is, Michael, it's yes, that
That's
we
right.
do.
And that would be a different mechanism. That would be with EpicNet, where NIH, through the HEAL initiatives, has set up a phase 2 clinical trial network for pain studies. Okay, so if you put into your browser EPICCNET, EpicNet, NINDS, or NIH, and then you'll see all the details on that. And the program lead for that is Barbara Karp, K-A-R-P.
Yeah. And I can say that probably the best, most appropriate time would not be at IND. You probably need to have some phase 1 information to be ready, which is exactly what this program is trying to do as well, which is get stuff ready for EpicNet to hopefully get the phase 1 information. I am studying non-opioid analgesics, how to decide to go with this mechanism or a regular RO1 under the HEAL.
Email Michael Oshinsky with your project, and then we'll talk about it.
Exactly. I mean, email either Michael or I, and we get happy to talk about it. But
You got it.
This is
This
it's very
is
independent on what you're exactly proposing, and we'll have
That's
to
right.
see.
This is the optimization stage projects. If you're considering doing it in an RO1, you're probably before this mechanism. But again, we could discuss it.
Is CPP data sufficient for demonstrating non-addiction for this application? Again, it depends on
any data you have is good, right?
Whether
or not
stage you're coming
sufficient
in.
is going to be determined by the reviewers and program staff. We just have to look at it.
But if you were coming in and saying, I have a development candidate and I want to go directly into, IND enabling tox studies, I would say you would need to have more than CPP. If you're saying as part of the optimization.
But if it's a mu receptor agonist, again, it's not our priority in this program.
Yeah, well, that would be non-responsive if it was a mu receptor, as I mentioned earlier. But okay, if we have
we're looking for outside the box and creative use of the other targets for pain.
Okay.
That's really
if we have a mechanism that involves mu opioid receptor, but have data indicating that the lead is not addictive, but no reward by CPA, no bar pressing by self-administration.
It's the same question. Got it. I mean, we can say it over and over again. Mu opioid agonists, not going to be a priority for this program.
I am working in India. Am I eligible for a grant from NINDS? You are eligible to apply, and if you meet all the rules, you're judged by the peer review to be a fundable application. There is an extra step for a foreign institution, as Michael alluded to, that we have to go through and go to council approval. And if you contact me, I'll send you the link about foreign institutions and the process. If the sponsor has an existing BPN UG3 grant for other routes of administration, would this impact the decision to fund? It might, is the answer to that question. It would really depend on the details that we were talking about.
Contact us.
Contact us. Are there different strategies that a researcher would use in drafting specific aims for an SBIR, STTR grant application versus RO1s?
Yes.
Yes.
Again, yeah. For those details, I mean, the development of a product is a part of the discussion for an SBIR grant, whereas when it comes to RO1s, that's typically not a part of the review criteria. So you have to have a business plan in an SBIR. You do not need that for an RO1, and that's only one example of many other things that are different.
And in this particular UG3, UH3, it's also different from the RO1 because we have to have a plan to get to a product and a patent strategy. And
you also have milestones in the cooperative agreements, and there's no milestones in the RO1.
So there's lots of differences. So again, if your best thing to do when you have those questions is contact Michael or I.
But you also don't have access to any of the contracts that were mentioned in Dr. Cywin's presentation on an RO1. None of them.
No.
But on the cooperative agreements, so the actual amount of money NIH is spending on these projects is much more than the budget that you put into the application because of the access to those contracts. And you don't get that benefit in an RO1.
Yeah, the contracts are cited.
And the consultant expertise, too.
And the consultant expertise, and our time is actually usually much more than the actual grant cost.
You're going to want to take that WebEx reminder off the screen there.
Oh, sorry. Didn't you see it pop up?
Sorry.
I'm looking, I have two screens. I'm looking at the questions. The other screen.
Yeah, I got it. Yeah, sorry.
I thought I had my screen paused, but I guess I don't. Let me repause my screen so that doesn't happen again. Concerning NINDS has established contract support for PK, tox, non-safety testing, provide access to experts in therapeutics developments through consulting service. Should we include somebody? So everything in there is correct, but the last part of it, can provide access to experts in therapeutic development. When I say therapeutics development, I definitely want to make sure I emphasize not disease biology, but therapeutics development through the clinic. Should we include those in budgets? No, you don't include that stuff in your budgets. But if you have, as Michael said, a collaborator for clinician, a collaborator for disease biology, pain expertise, that should be part of your budget. What are acceptable sources of matching funds? Matching funds is not required.
I think you might be referring to your suggestion that this not be the only funding for your lab. So that doesn't have to be a part of that was advice about your company and how to use these funds.
I think it's actually a reference probably to the previous FOA where it was required. And then
that might be possible. Yeah, that's true. The optimization, yeah.
We have a molecule that has undergone phase 1B studies in Europe and can submit an application under this program. Can we submit an application for improving the formulation and submitting an IND? But yes, potentially you could. I
don't see why not. It's definitely take advantage of our contracts.
That would be a development project.
Yep, development project.
And you could potentially do that with your own contracts or through our contracts, depending on what exactly the entity is. I am founder of a company based in New York, and we're developing a non-opioid pain medication. However, the majority of investors are not U.S. based or green card. Keep going, Tim. Yeah. Keep going. I can't see the bottom of this question. Holders or citizens, is this funding opportunity open to non-U.S. citizens? Yes.
Yes.
Do we need to collaborate with a pharmaceutical company? Not at this stage. We obviously are hoping down the road you will collaborate to get your project further into phase 2 and beyond. Whether that be venture capital or pharmaceutical company or some other means, we hope that you continue to do that. Is a topical cream based on nanoparticle technology for pain treatment included in this program? Could be. Again, devil's in the details, but acute pain is part of the program. Will it fund phase 1 clinical trials? Yes. Like I said, the plan is to fund that through our contracts mainly, but you can propose that as well through your own contract work. It would then become a clinical trial application. But I need to say, you cannot just apply to do the phase 1 clinical trial. You can't come in and say, I have everything, I just want to run a phase 1 clinical trial. We have another FOA for that. If you just want to do a clinical trial, you have all the data ready to go.
Right, so reach out to us if that's the case, and we'll guide you to the appropriate funding announcement for that.
We should not apply to, my understanding, we should not apply for RFA 21-010, which I'm not sure, and Ignite with similar aims at the same time.
That is completely against the rules. Yes.
Correct. Yeah. You can't apply to any 2 funding opportunities at the same time with the same aims.
That means both applications do not get funded. That's what I mean by it's against the rules. If you score well on both of them, it's not like we pick one. We tell you you broke the rules and you don't get either one.
Right. So the bottom line is until you get the summary statement from the first application, you can't apply with those aims again.
Until you get that summary statement.
Right. You have to have the summary statement in your hand. Is the project more likely to be funded if it proposes to use NIH contractors? No, it's not. I don't believe that to be the case. But again, it has to do with total amount of your budget, whether you are able to do this, get up and running at the right away. You don't need to then go off and contract later. Can you handle the management of those contracts? All that stuff you're going to need to at least put in your plan how you're going to manage all that stuff. But
if you just need money, then there are other funding announcements to take advantage of. The benefit of this program is to have access to the consultant expertise and those contracts. If you just need money to do the project, there are other funding mechanisms that you can use, and you can reach out to me, again, Michael Oshinsky, and I can help you with that.
Yeah, but there will be cases where you'll use your own contractors for, especially in the biologic space, where we may not have a contract in place for you. And we'll expect you to do that through your own contractors, and we can potentially help you with our consultants, where the consultants can help you is in managing some of that stuff as part of the team. How about peripherally restricted opioids? Again, if it's maybe opioids, the answer is no. Is the submission November 2020 or 2021? 2020 is the first one. So, whoops. What is the budget limit for all 5 years? You budget what you believe you need. Is that it, Tim, for questions? Have I reached the bottom? I probably reached the time almost.
No, there's a question. Do you have a contract for pain animal efficacy models? If all you need is animal models for pain and you have a potential small molecule therapeutic or a biologic, we have another program for that at NIH funded by HEAL. It's called the PSPP program, and you can reach out to me or to Smriti Iyengar for that information.
Yeah, but the answer to that question is I do not have contracts for pain models that can drive within this program. So if you're in this program and you're asking about running an SAR program, PSPP can't do SAR studies for you.
No, that's not what they're for. They're for testing,
right?
And
so
and some profiling.
Right. So
it's a preclinical screening platform for pain, PSPP.
You need to put together a team, get collaborators to do that on a regular basis, if that's what you're asking. Will cell therapy or gene therapy also be considered?
Yes.
Yes, they will. And Marie, would you like to comment?
Yeah, please.
Yes, absolutely. It's part of biologics.
Okay. No prior arrangements with NIH contracts are required for applications. No, none are. You just need to say what you're going to do. You're also what you need to do. And like I said, talk to us early about that so that we make sure you're not proposing to do something we can't do. Formulation development, then IND tox could be responsive. You'd be saying you're coming in as a development candidate. If you have all the preliminary data that backs up the program, the tox data that backs this up, meaning you've got dose range finding studies, you've got everything you need to get ready for IND tox, then yes, it could be.
Okay, that we are at the end of the question. We'll give a couple more seconds to see if we get any more questions, and we do. Is lasers technology considered biologics?
No, that's a device. That wouldn't be appropriate for this program. We have other programs for devices, so feel free to reach out to us.
Mm-hmm.
Okay, I think that is going good. We will send out, someone asked for Michael Oshinsky's email. I will send out an email to everyone with everyone's contact information. In addition, everybody will get a copy of the slides and a recording of this webinar.
But Tim, do me a favor, just add my name to the slide under Chuck's name for an INDS. That might be helpful for everybody. Okay. You can reach out to me or Chuck.
Not sure what just happened there. I lost my thing. It switched around on me. And there we go.
Hey, Chuck, there's one other question that just came out, if you can read it. You see it?
You mean the formulation development question? Oh, no. Even though it affects biological systems through non-thermal means, yes, it's a device. You're developing a device, if that's a question.
The TRPV1 question?
We have we have a TRPV1 receptor antagonist that went through phase 1B trials in the EU. Can we apply here for a new formulation IND? You can, like I said, again, the devil's in the details. If this is going through an IND pathway, then chances are yes. You would have to say why you want to do it. So on TRPV1, obviously it's been looked at a number of times, so you need to put it in context of why what you're doing is important.
Okay, I think with that, we're going to end. So thank you everyone for joining. And we'll, like I said, everyone that has registered, I will send out the recording and the PowerPoint slides, okay? So thank you to everybody.
Thanks.
Thanks, everybody.
Thank
Great
you.
questions. Yep, be well.